Now we're going to look closer at antibodies. So antibodies are small kind of proteins. They're soluble, meaning they dissolve in water, and so then they're in the plasma. That's how they circulate the body or the lymph. And again, they're specific. They come from specific B cells that lead to specific plasma cells that make specific antibodies that'll fight against those antigens. Now we're talking about abundance. The more antibodies, the more we can fight against the extracellular pathogen, whether it's a bacteria or a virus. And so we get a better response later, which we're going to talk about in the next video. So we look at it two polypeptide chains that are next to each other. So we have a pair of heavy chains and then light chains. Most of the time we have a constant segment. That's going to be what type it is. So we're going to have different types of antibodies. So we're going to have IgG, IgM, IgE. So the constant segment is going to determine that. The variable segment is going to be based on the antigen it's fighting against, and what we call its antigen binding sites. So this is where you can have the specificity of the antibodies, is where the specific antigen will bind to its specific site. So this is just looking at it. We have two antigen binding sites. This is why you've seen me kind of draw it in the past that it looks like a Y. So there's two antigen binding sites, which is your variable site. And then we determine the constant, which is, again, the type that we have. So when the specific antibody binds to its antigen, we call it an antigen-antibody complex. So again, if this antibody is going to be finding its antigen specific, then it's considered the antigen-antibody complex. And a lot of times this is going to be on the surface of a bacterial or viral cell. So what happens is it's going to be a specific part of the antigen called antigen determination sites or determinant sites. So again, a bacteria may have many different things on the surface of its cell that's going to be able to be recognized by B cells and T cells as well. So what happens is then we can produce antibodies to these specific sites. So an antibody will match this one, an antibody will match this one, an antibody will match this one. Once it's bound to its antigenic determinate sites on the antigen, then this is when it's the antigen antibody complex. They're bound together. So that's why I say bacteria can contain millions of antigenic determinant sites where these antibodies can attach to. So you can have millions of antibodies covering a bacteria so that we can take care of it. And again, the immune system, the other part of the immune system, mainly macrophages, can take care of it. So again, antigens are going to be anything. So antigens are substances that can provoke or start an immune response. So again, it doesn't have to be the entire bacteria. It can be one little piece of it. So that can be an antigen. Complete antigens, we consider ones that have two antigenic determinant sites that can have one for each binding site of the antibody. So if we have two, then the antibody can come in and be it's kind of Y-shaped and can attach to these two sites. So we call that a complete antigen when there's two sites for the antibody to bind to. Haptens we call partial antigens, because normally they can't cause B cell activation, because again, if you only have one of the antigenic determining sites and not two, well, the antibody can't attach. It can't have one attached and then one free floating. That doesn't happen. Sometimes when we do have a response to haptens, they actually oftentimes are toxins. So haptens, toxins that they bind to a carrier molecule. And the carrier molecule has a matching, so it can cause a complete antigen. So they will attack both the hapten and the carrier molecule. This can be dangerous because if the carrier molecule is normally found in your tissue, so it's a normal cell part, then your antibodies may start attacking your normal cells that should be there. Oftentimes, haptens can be toxins, but also drug reactions. So penicillin reactions can be attacking normal cells because you're attacking the carrier cells for the penicillin. So this is just showing you antigenic determining sites right here on your bacteria. That makes it a complete antigen. The antibody can bind to it, making an antigen antibody complex. Hapten, so if you just have this, well, it can only go there, but it's not two sites. So you have the carrier molecule that the hapten attaches to, and then you have two antigenic determining sites. So the antibody can bind to it. So that's the major difference. So again, a bacterium or one single bacteria cell can contain millions of antigenic determining sites. So it kind of looks like a potato here, sesame seeds. So all of these little seeds are going to be antigenic determining sites. So as long as the antibody can find two of them that it can bind to together, then it can bind. So this is an antigen antibody complex because we have antibodies attached to the antigen. So like I said, this is going to be classes of antibodies. So they have the same kind of constant. So they have the same constant chains, but then they will have different variable chains. So they still have different antigens that they'll fight against, but they're in the same class. So IgM is going to be our largest. So it has actually 5 Y-shaped monomers. So the way IgM is got 5 Ys in kind of an arrangement like that. This is the first antibody that's released. So again, B cells are activated. They clone themselves and then differentiate into plasma cells. The first type of antibody that plasma cells will release are IgM, the largest 5 pentamer. So you can think of it as pentamer, 5 is pent. This is going to be an agglutinating agent. And so agglutinating is making clumps. They also activate complement. So remember the proteins that will help fight against infection themselves and then communicate between other parts. That innate or non-specific part of the immune system, it'll help activate that as well. So again, it's communication between your specific and your non-specific. So why is IgM a significant agglutinating agent? Well, you can kind of make a big clump of these. So if you have the 5 Ys and it attaches to kind of antigens here, well, then this one can clump and attach to antigen over here. And then these guys can be another one, and then there can be another clump and another pentamer. So you can just make a big clump of these guys. And so that's why we say an agglutinating agent. So you just make a big clump. The clump's easier for macrophages to come along, and then eat the big clump. And talk about agglutination a little bit more. IgG is the more classic one. So it's the one that we kind of draw that it just looks like Y-shaped and that's it. It's just one Y-shaped. This is going to be the most numerous or the most abundant antibody that travels around your body. So this is going to be, again, traveling around in your plasma. So 80% of all antibodies are actually this class of IgG. They are small. So look at the difference between 5, a pentamer of IgM kind of stuck together versus just 1. So they can actually enter tissues from the blood vessels just by slipping through because it's a smaller protein, smaller molecule. IgG can fight against bacteria, viruses, and toxins. Same thing that we saw in IgM. It can trigger complement. So again, those proteins that fight against infection and coordinate the immune response as well. And this one is the only one that can cross the placenta. So again, it's small, so it can go through the placenta of a pregnant mother, and it can have that passive immunity. Remember, antibodies directly to the newborns. So it can go directly to the newborn and help fight against infections, especially early on in life. IgA is a different class, and it's going to be 2 Y-shaped monomers, so not as large as IgM, but larger than IgG. This is going to be cells in your mucous membranes, so things that are opening that are kind of pink membranes, so things like your mouth or oral cavity. This is important. So if you have a lining, so the lining of your mouth, you have these cells that are going to have IgA on them or produced by them. So they'll kind of help prevent bacteria and viruses from attaching to the surface of the mucous membrane and then traveling into the body and getting there. So what happens is they can stop the pathogens before they can get inside. So they are found in areas like mucous membranes conjunctiva, so around your eye. So they're found in saliva, mouth, tears around your eyes, sweat or perspiration. So again, this is going to be where all our barriers or first line of defense are. Colostrum is going to be first breast milk. So another protective way for newborns. Mucus that's made by respiratory tract, especially nasal cavity, things like that, and also semen. So they're found in many different places so that we can stop the pathogen before it even gets inside the body. So it's all at the entrances or those walls or first barriers into the body. IgD is going to be another Y-shaped monomer, but it doesn't circulate. So it's a little different in the fact that it's the receptors on the B cells. So IgD is actually the receptors that are on the B cells that then create differentiation for the B cells to become plasma cells to make more. So they're small in the Y-shaped monomer, but they're specifically attached and associated with B cells. They're not circulating. So that's the major difference. IgE is Y-shaped, but a little larger Y. You can think of it as a capital Y versus a lowercase y. This is going to be attached to mast cells and basophils. So remember, these guys are associated with inflammation. So remember, mast cells and basophils have granules filled with histamine, which is our major inflammatory chemical, which causes that vasodilation increased blood flow to the area, can cause swelling, pain, all of those signs that we see for inflammation. So these guys, IgE is often associated with allergic reactions because a lot of times that's what's happening. The mast cells or the basophils may be reacting to a pollen or a skin cell from another animal or a dander or whatever, and it releases histamine. So histamine, again, allergic reactions are basically inflammation to kind of things that you wouldn't necessarily think we need inflammation against. You don't necessarily need to fight a pollen molecule from a flower or tree. So it's inflammation kind of unnecessarily. It also is associated with parasitic worms. So they are also associated with parasites and eosinophils as well. So, how do antibodies fight? So, these different classes of antibodies fight against antigens. First one is neutralization. So, what happens is they can attach to receptors on the bacteria or the viruses and block them. So, normally, say a bacteria needs this receptor to infect a cell and go inside. Well, if there's already an antibody attached to it for kind of what it's supposed to, then it's going to try to go in the cell and it can't. So it's neutralizing its effect, it's taking away its weapon. IgA, specifically in those mucous membranes or secretions around glands like salivary glands and the lacrimal gland, can stop bacteria, and they oftentimes on kind of those barriers, so the oral cavity specifically, are going to attach to the mucous membranes and go into the body cells, but IgA is going to prevent it from sticking originally, so it can't even do its job right off the bat. We talked about a couple of them, like IgG activating complement. Remember, complement can also come in and it can produce its killing proteins and communicate with other parts of the immune system. So activating complement is important in helping in fighting against specific bacteria and viruses. IgE is going to be the major one of this one, of stimulating inflammation if we need to. So it's going to stimulate basophils and mast cells to release chemicals. Majorly, it's going to be histamine, which then will lead to inflammation. And remember, inflammation isn't necessarily a bad thing. It's going to help more white blood cells to come to the area, keep it immobilized so more white blood cells can have time to do their job. So it's going to help with fighting off an infection. Opsonization is going to be a common one. Complement also does it too, so proteins can attach to it. Basically, this is going to be kind of the one that I've been drawing a lot. So you have the pathogen and you have all of its different kind of receptors that are recognized by the antibodies. They're just going to come along and just code it completely. So this bigger kind of complex of antibody antigen complex is more easy for, especially again, the macrophages to come in and eat this whole big meal versus a bunch of little meals. So it's easier for this to be consumed by the phagocytes. So that's what opsonization is. It's just kind of a coating to make it easier for phagocytes to come. And they also, antibodies are going to attract eosinophils, neutrophils, macrophages. What are those? Those are all your phagocytes. So it's going to attract these white blood cells to the area so that they can eat and eat all of these kind of coded pathogens that they need to eat. Antibodies can also agglutinate or precipitate. So showed this too, where they can have a big clump of multiple pathogens by the antibodies. Sometimes they become insoluble, meaning they're not dissolved anymore. So they're not in solution. So what happens is they precipitate. So it's sort of like if you all of a sudden have a cup of sugar water and you've mixed it, so it's all kind of mixed around. And then all of a sudden you leave it, and then you have a bunch of sugar granules at the bottom, salt, anything. So you have precipitation where you have actually solid things. That precipitation, more often as well, if it's insoluble and it's not dissolved anymore, phagocytes can eat a little easier, so they can consume easier when it's precipitated. But we also have agglutination, which we talked about when we looked at cross reactions for red blood cells, so blood typing. We saw agglutination, so you saw a big clump of red blood cells. That was from the antibodies. So again, antibodies are going to come and they may attach to that guy, and then they may attach to another one and make a big clump. And then this one, kind of IgM, will clump to this one, and then this one will clump over here. So we just make a large clump of antibodies and antigens. So agglutination is a big clump. So you can see just kind of better neutralization is going to be where you're just kind of similar to opsonization, where you're just surrounding it. So they can't have an effect. You're taking away the bacteria weapons. They can't attach. They can't go into cells. They can't do anything. So you're just taking away their weapons. Agglutination, again, is a clump. So this is showing you red blood cells. So they're all kind of clumped together in a big, large clump. So antibodies are surrounding the antigen. There's a bunch of antigens with the antibodies. And oftentimes this is an IgM. So you see this. Precipitation, again, there's going to be kind of antibodies come in and they do make a big clump, but they just come out of solution. Again, all of these are going to, the antibodies are going to trigger complement. Remember, complement makes a membrane attack complex that can help kill the cells as well. And it can also communicate with the rest of the immune system. So all of these are going to enhance phagocytosis by those phagocytes. So the neutrophils, the eosinophils, and the macrophages. Complement is going to also, and antibodies are going to enhance inflammation. Inflammation is also chemotaxis, so we're going to bring more phagocytes to the area if we need to. And so we're going to have histamine release, and that allows more time and more cells to come to the area. And then we also have cell lysis, which is going to be majorly complement for that membrane attack complex. So if we need to, we're going to pop the bacteria cells, destroy the viruses. So it's all kind of, again, working together.